Today's discussion will be about the vestibular system, which is involved with our balance and our ability to keep things in focus while our head is moving. The vestibular system is part of the inner ear. The external auditory canal and the middle ear are involved with the cochlea, and this is for the perception of sound, which will be a different discussion. But up here you can see we have the semicircular canals embedded in the bone. So we have a bony labyrinth and we have a membranous labyrinth. Let's look at the vestibular part in more detail. Here we have that part of the labyrinth. It really does look like a labyrinth, like a maze here, composed of the three semicircular canals. And then there is a larger vestibular area, which has two areas involved in sensing gravity as well as linear acceleration. And this is the utricle and the saccule adjacent to this area called the vestibule. Now, these membranous canals have sense organs inside them, and the semicircular canals are particularly involved with sensing angular acceleration, and these canals are oriented in three different planes so that all three dimensions of your movement are registered. And when we test the vestibular system, we are almost always testing the horizontal canal. It's easiest to test, especially if you bend the head forward about 30 degrees. So this will be our focus today, and let's look at another diagram. Here we have the membranous labyrinth in blue. This is filled with endolymph, and at the base of each of these semicircular canals we have a region called the ampulla, where the sensory hair cells that are going to be responding to angular acceleration are located. And in the saccule and utricle, in the vestibular area or vestibule, we have the sensory hair cells that are going to respond to linear acceleration and gravity. We will look at those hair cells in more detail, but first let's look at the skull and look at the orientation and location of the labyrinth with respect to not only the cochlea, but to the vessels and other structures on the internal surface of the skull. I have here a skull, and we're looking at the inside of it, and it has been specially prepared to show you the vestibular and cochlear areas of the bone. The petrous part, this part here, and the Petrous Ridge are part of the temporal bone. And if I tilt this, you can see the internal auditory meatus through which the seventh facial and vestibulo-acoustic nerve pass. And the structures we are interested in are deep in this bone. And this area has been dissected from the outside, and we can look at that. Here we have the mastoid process. You can see the air cells, and deep in the bone you can see one of the semicircular canals. And right beneath here is the foramen through which the facial nerve passes after it leaves the temporal bone. Now let's look at this from the inside and look at our landmarks. We have the vessels, which are most obvious. The middle meningeal artery comes up and splays out over the inside of the skull. It's really running in the dura, and it is right under this squamous part of the temporal bone, which is often damaged or lacerated in trauma cases and is the source of epidural bleeds. We also have the blue venous drainage. We can see the sinus coming along here and then going down and entering into the jugular foramen and returning the blood from the brain to the heart. The Petrous Ridge here has been partially dissected. The yellow here represents the facial nerve. Here is one of the semicircular canals, and up here is the cochlea, the little tiny structure involved with the perception of sound. So now let's look at the temporal bone in an isolated section, and you can see how complicated it is. There's the mastoid process, the zygomatic process. This is facing anteriorly, so this is from the left side of the skull. This is the squamous part, the more delicate part of the bone that can be easily lacerated in trauma. And as I turn it around, you can see the relationship again of the internal auditory meatus, where the vestibular nerve is running, and you can see the vestibular apparatus, one of the semicircular canals. Now let's look at the membranous labyrinth in detail from a large model. This giant model of the right ear and the petrous part of the temporal bone, which I can remove here, will expose for you both the cochlea and the semicircular canals. So let's just lift out and separate the semicircular canals and the vestibule from the rest and look at it in a little bit of detail. So now I'm holding the labyrinth, and you can see that the white part represents the bony labyrinth, which is filled with perilymph. That would be the pink fluid, so to speak, in this model. And I can lift out the inner membranous labyrinth that is filled with endolymph. And that is the fluid that swishes around in these canals. I only have one to demonstrate to you, or moves around in the utricle and saccule and stimulates by its movement the hair cells. The semicircular canals have an ampulla with hair cells, and the saccule and utricle also have hair cells, and we've already mentioned their specific functions. And you can see the nerves coming from each part and forming part of the vestibular nerve. Now let's look at these hair cells that are the transducers of the movement of the endolymph that become the signal to the brain of position movement. Might mention that this labyrinth can become infected. We call that labyrinthitis. And labyrinthitis is an inflammation which usually is due to viral or bacterial infections. And the patient has nausea, disorientation, vertigo, and dizziness when you interrupt the mechanism for transduction. Here we have the hair cells in the macula, and each one of them is innervated by an axon of the vestibular nerve, and each of these hair cells has one tall cilia called a kinocilium, and then many smaller, shorter cilia called stereocilia. And it's very cleverly designed so that whichever way these cilia bend, if they bend toward the kinocilium, the nerves are excited. If they bend away from the kinocilium, then the nerve is inhibited. And with this mechanism, we have a mirror image in each ear. So either one side is stimulated. If that's so, then the other side is always inhibited. Now, on top of these hair cells is a membrane. We usually just call it the otolith, with crystals of calcium carbonate that are sort of embedded in a jelly, and they're about twice as heavy as the fluid, the endolymph that surrounds them. So with the movement of the body, the kinocilium and the stereocilia are deformed and produce either excitation or inhibition. This mechanism is different than what we see in the ampulla at the base of the semicircular canals. Let's look at that. At the base of each of the three semicircular canals is an ampulla, and at the base is also a crest here, which we call the crista, with hair cells along it, each again innervated by a vestibular axon. These hair cells have the same arrangement with a tall kinocilium shown here, and the other stereocilia are not shown. These stereocilia and kinocilia are embedded in a gelatinous cupola, and when the fluid, pretend that you imagine fluid in this space, moving, they deflect the cupola, and if it's in the direction of the kinocilium, then the nerve discharges, and if it's in the opposite direction, it inhibits. And since each of these on either side is a direct mirror image, if you stimulate the right side, then you are inhibiting the left side. And let's talk just about the horizontal canal. So this is the anatomy of the cupula, and this fluid is always sort of lagging behind the movement of the body. Now, we need to bring this nerve here into the brain, and so let's look at where the vestibular nerve enters the brain at the junction of the medulla and the pons. Here I have the ventral surface of the brain stem and cerebellum that have come out of the posterior fossa of the skull. You can see the medulla and the pons. And out here in the cerebellopontine angle, you can see two nerves, the more medial facial or seventh nerve and the more lateral eighth or vestibulo-cochlear nerve. That nerve actually has two parts. You can sometimes see a groove in it. And we are going to be talking about the connections of the vestibular system with nuclei in the brainstem and tracts that go down into the cervical region. So the important part of the vestibular system is in the brainstem, not in the cortex. In this angle here, called the cerebellopontine angle, sometimes tumors develop right in this space. There's just room enough for them to grow, and these tumors are often on the vestibular or cochlear nerve. They often involve the seventh nerve, and they come from Schwann cells mostly, or sometimes from the meninges. Now let's look at a cross-section. So this is a section through the medulla with the cerebellum on either side, and you can very nicely see the vestibular and cochlear nerve coming in right over the inferior cerebellar peduncle. And there are some connections actually that go to the cerebellum from the vestibular nerve, but most of them end up in some nuclei right here called the vestibular nuclei. There are 4 of them, and they extend all the way from the medulla through the pons, and they have very important connections for controlling eye movements. And we're going to look at some diagrams indicating that in a moment. So what I'm going to do now is looking at the superior or dorsal surface of the cerebellum. I'm going to split the cerebellum open, and now we're going to look down into the floor of the fourth ventricle. So now I have split the center of the cerebellum, and you can see the vermis on either side. And this is the beginning of the midbrain. And so now we're looking down onto the floor of the fourth ventricle. And our vestibular nuclei are going to be located right near the midline on either side. Let me remove the cerebellum now. So now we have a bit of cerebellum, but we can look down very nicely onto the floor of the fourth ventricle and imagine where these nuclei are. There are 4 of them. We don't need to know their names, but they have connections with tracts that ascend and descend, because after all, we have to coordinate neck movements and eye movements with our position in space. Very important, the vestibular nuclei. So in this mid-sagittal section, we see the whole half of the right hemisphere, and the brainstem begins here at the aqueduct and the superior colliculus and extends down even a little farther than you can see here in the medulla. So the only part of the vestibular system we're going to talk about, although there are connections with the thalamus and the cortex, for diagnostic purposes, it's the brainstem that's important, and we're going to be looking at in more detail, and I'm going to show you a tract that runs in this dimension here. This image shows the vestibular nuclei, the 4 vestibular nuclei, in a computer-generated graphic. So let me orient you. Down here we have our classic cross-section through the medulla, and up here are section through the pons, all the way up to the rostral part of the pons. And so the vestibular nuclei extend all the way through the pons and the medulla. This is important. And they give off branches to a tract that runs in the midline on either side. This tract is called the medial longitudinal fasciculus. The medial longitudinal fasciculus, or MLF as we call it, is what connects the cranial nerves with the vestibular system. Let's look at that now. Here we have another computer graphic, and this time showing the medial longitudinal fasciculus, that tract to which the vestibular nuclei contribute axons. And this green tract runs all the way from the superior colliculus at the level of the oculomotor nucleus all the way down to the high cervical level for coordinating neck muscles and the position of the head with respect to the object that you want to focus on. And disease of this MLF is very pronounced and results in a variety of changes that we're going to discuss momentarily. So the MLF, a highly myelinated pathway that extends all the way through the brainstem. That means it's also a useful pathway to test for the integrity or the viability or the functioning of the brainstem. So now let's suppose that we have a lesion of the left vestibular nerve. What would happen, or what would the patient report, or what would you see? So let's imagine that this nerve, and we're just now talking about the horizontal portion basically of the semicircular canal, because that's the one that's easiest to understand. We would have a disparity in the information coming to the vestibular nuclei on the left side when compared to the right side. And so since movement on this left side is normally balanced by inhibition of the other side, that input is no longer there. And so the right side thinks that it's always moving. And so if you have your right side moving to the right, so the brain thinks your body and your head is moving to the right, you want to keep the object in focus, so you slowly move your eyes to the left. You can test that on yourself. That you have something in focus, you turn your head, and your eyes go in the opposite direction of the head. And that is a slow phase, but pretty soon you end. Your eyes can't go any farther to the side, and so then you flick them back from this mid position out to the side, and you have a fast flick back, and that is called a saccade, a fast movement. And so we have a condition where we have jiggly eyes, so to speak, in which there is a slow phase and a fast phase. And we call this movement of the eyes, when you're not moving, nystagmus. And nystagmus can either be pathological or physiological. In this case, we've given you an example of pathological. Nystagmus is a complicated thing to understand. It can be in any direction. It can be up, down, roundabout, and it can be congenital or it can be acquired. The most common cause of nystagmus, acquired nystagmus, are drugs, medications, such as dilantin or phenytoin, used for controlling epileptic seizures. And excessive alcohol can cause this, or certain sedating medicines. So nystagmus is not normal. If the person has it, then you need to search for the cause. So the cause could be in the nerve, or the cause could be toxic. And we want now to go on and discuss the role of the vestibular system in what we call the vestibulo-ocular reflex. So this is a reflex that happens when you turn your head, in this diagram, to the left. So we know now that if the head goes left, the eyes should turn to the right. But what is the connectivity for this? We've already mentioned the medial longitudinal fasciculus that runs all the way from the medulla up to the midbrain. But we have to coordinate cranial nerves, 6, the abducens, and 3, the midbrain, oculomotor nucleus, and they are on opposite sides. So this is a complicated diagram, but many of the other diagrams you'll see are in error. So pay attention to this one. We're going to stimulate now the semicircular canal, the horizontal one, on the left side. So this is the left vestibular nerve coming into those vestibular nuclei. And what is not very logical is that the next neuron here in the vestibular nucleus crosses into the MLF of the opposite side and travels up to the right abducens nucleus. And in the right abducens nucleus, there are 2 types of neurons. The logical one, which you need to turn your eyes to the right, are the motor neurons that come out through the abducen nerve to the lateral rectus muscle. But these other neurons, interneurons here, go up to the 3rd nucleus, but it has to be the 3rd nucleus on the other side. So these neurons cross and travel up through the MLF on the opposite side, on the left side, to terminate in the oculomotor nucleus. And there, motor neurons from the oculomotor nucleus travel out on the left side to the medial rectus, so that you have coordination and you can track to the right when your head is turning to the left. Now this reflex is important because look, it is testing from the end of the pons where the vestibular nerve comes in, the junction there between the medulla and the pons, all the way up to the superior colliculus, and that's a couple of inches. And so this is a great test for the integrity of the brainstem, for the pons and the midbrain, and it's useful and very important clinically. So damage to this MLF, medial longitudinal fasciculus, will usually result in nystagmus because you have an unequal stimulation on either side. And we are going to look at something called an internuclear ophthalmoplegia. This can be from a disruption of the MLF. It could be on either side, and it could be anywhere from the pons to the superior colliculus. And with it comes nystagmus. It's different from the vestibular lesion nystagmus, but it is a place where you see nystagmus, and it's often associated either with a small brainstem stroke or quite often with demyelination of this tract in multiple sclerosis. Here we have a diagram of the pupil of a person who is looking straight ahead at you. This is normal, and they are fixed on you, and you command them first to look to the right down here, and both eyes move to the right. So here you're using the lateral rectus on one side and the medial rectus on the other side, and there is no problem. You then ask them to quickly look to the left, and what happens is the left eye goes over, so there's no problem with the left abducens, and the right does not go over. The right actually will move a bit, but it doesn't go all the way over. And if you ask the patient to look at your fingers to see if they can focus on something, you'll see that both eyes can converge. So it's not a problem with the medial rectus muscle. And that puts the problem in the MLF that connects the nuclei 3 and 6. 3 doesn't move all the way over. This is more or less paralyzed. And this eye moves all the way over, and then it starts to oscillate. It starts to jiggle. You have nystagmus. And this is abnormal. It's as if all the electricity went out the abducens nerve because no stimulation is going to the oculomotor nerve. That's not necessarily true, but it's a good way of thinking about it. And this condition is called internuclear ophthalmoplegia. Internuclear between the nuclei of 6 that go to the lateral rectus and the nuclei of 3 that go to the medial rectus. And so what we have here is a unilateral internuclear ophthalmoplegia, a paralysis between the two nuclei, 3 and 6, producing paralysis. And in this case, we have, since it's the right medial rectus that isn't working, we call this a lesion of the right MLF. Now let's look at a patient that has this condition.
This patient had subacute bacterial endocarditis with a bacterial abscess in the brainstem. Abductions and gaze are fairly full looking to the right, but look at this. When looking to the left, his right eye does not adduct well, and you can see these jerk abducting nystagmus of the left eye. So he has a right internuclear ophthalmoplegia. Again, the components are medial duction abnormalities of the ipsilateral eye and abducting nystagmus of the contralateral eye. Now these are really brought out with saccades. This is a nice technique to bring out an INO. Ask the patient to saccade quickly to the right, as he's done here, normal, to the left. Now look at the lag of the medial duction of that right eye. Here, see how it just sort of slowly drifts across. That's a nice way to bring out a subtle INO. I think it shows up fairly nicely. Look at the left eye, you can see the abducting nystagmus, and that medial duction of the right eye just slowly drifts across. You can also do this with an optokinetic drum or strip by moving the target opposite in the direction of the suspected gaze. Now here's another example, fairly normal when looking to the left. Now look at that left eye medial duct slowly compared to the right eye. And again, I think you can see the prominent abducting nystagmus of the right eye. This patient has multiple sclerosis.
I'm going to use myself as an example of how you could test on a conscious person whose cerebral cortex is working and demonstrate the vestibulo-ocular reflex. And I'm going to spin around on this stool, and it's going to stimulate the endolymph in my horizontal canal. And when I stop, my eyes are going to flick back and forth, and I'm going to have nystagmus. So let's look at that now. So now I'm going to spin around, and I'm turning to the right. So normally my eyes would want to track to the left. And now I'm going to look at the camera, and you should see my eyes jiggling. That's nystagmus. That's physiological nystagmus. That's normal. So now, finally, I want to talk about caloric stimulation of the semicircular canals. That is irrigating the external canal with either warm, around 44 or a little higher degrees centigrade, or cold, about 30 degrees centigrade. And what happens when you cause, with this heat or cold, the endolymph in the canal to set up convection currents and artificially stimulate the hair cells in the cupula, and in this case, the horizontal canal. So the body is not moving, but the brain thinks you are moving because you're a conscious person, your cerebral cortex is working, and so the irrigation then causes my eyes to move, and there is a mnemonic for remembering which way the nystagmus is beating, as we say. The fast component is the beating phase. So we have a mnemonic for caloric testing in an awake patient, and we say that it is COWS, cold, opposite, warm, same. That refers to the direction of the fast phase, the saccadic part of the nystagmus. Cold, opposite, meaning your eyes beat to the opposite side. Warm irrigation, the eyes beat to the same side. Now, when we have a comatose patient, the cerebral cortex is not working. So when we irrigate the canal and they're lying on their back and their eyes are up and looking straight ahead, you can irrigate, but you will And again, you will set up the movement of the endolymph, but since there is no focusing the component of nystagmus for the fast phase, you will only have the slow phase. And so with a comatose patient, if the brainstem is working, you will only have the slow phase, and the eyes will drift to the side. So you will only have the slow phase when the head is turned. And if you have put in cold water, the eyes will drift to the same side. If you put in warm water, the comatose patient's eyes will drift to the opposite side. And you can then move their head to looking straight ahead, let the eyes equilibrate, and test on the opposite side. If you get the slow movement of the eyes, that is good. That is good news. It means that the MLF and the brainstem, at least for eye movements, are working over the distance from the midbrain down to the beginning of the medulla. So that's a very good sign. If the eyes do not move, that is a very bad prognosis and indicates that the brainstem is damaged. So this concludes our discussion, our all too long discussion about the vestibular system, but it's complicated and clinically it's useful for testing. We've introduced the terms MLF, medial longitudinal fasciculus, which coordinates the eye movements from the midbrain all the way down to the pons, and we've introduced the role of the vestibular nerve in the vestibulo-ocular reflex. And we've also talked about nystagmus, a condition where the eyes are moving when they shouldn't be. So hopefully you'll be able to look at your patient and figure out what's going on. Is it in their brainstem, in their vestibular nerve, or have they been drinking?